Okie doke. Good evening, everyone. So Holly, I'll hand over to you now so you can share your presentation and get going. Any problems at all, please let me know.
Yep, so I'll just share my presentation now. So just let me know if you can see it.
Yeah, things are coming up.
Awesome, I will just start PowerPoint. So can you see that okay?
We can indeed.
It's just the PowerPoint, I'm not like sharing all my tabs.
Just the PowerPoint, great.
Okay. So hi, I'm Holly. I'm here as part of the Oxford Personalised Medicine Society. And I'm here to talk a bit about what personalised medicine is and how we already use it firstly, and kind of what the future of personalised medicine might actually be. So as an introduction to me, I'm a medical student at Oxford, and I've completed the first 4 years of my medical training, and I'm currently doing a DPhil, which is the Oxford equivalent of a PhD in cancer science. I'm funded by Cancer Research UK, and I'm working in the Wellcome Centre for Human Genetics, which is this building you see here. I also tutor in medical genetics at St Anne's College. So I'm here as part of the Oxford Personalised Medicine Society, who are a student organisation, and we do a lot of kind of interaction with students at the university to really highlight their involvement in research. So Oxford's great because there's lots of opportunities to be involved in research as a student, and we hold an annual symposium each year to basically allow students to give talks, submit abstracts, and talk about the things they're doing as part of their FHS projects, as part of extracurricular activities, and it's a really good platform for that. So firstly, I think is a really good place to start is actually to say what is personalised medicine, because I feel like it's definitely used as a bit of a buzzword. It can definitely be overused, but personalised medicine is effectively a switch from one-size-fits-all medicine to a patient-focused approach. So this basically means there's a focus on patient stratification. So you might, for example, change your treatment based on somebody's family history, their previous medical history, so how they've responded to drugs in the past, and then that might influence your future choices. And what I'm going to talk about primarily here, their genomic and genetic data. And you might think, oh, don't we already do this? And we absolutely do. A lot of personalised medicine, even in the NHS, which is a public healthcare system where a lot of the more expensive elements of personalised medicine, such as genomic screening, are definitely reduced due to financial limitations, we still actually do a lot of work where we will, for example, look at somebody's family history to decide what diseases they're at higher risk of. So you might see this with things like heart disease, where family risk definitely affects screening for that. So you can see in this figure, basically how these people who have the same diagnosis and would normally be given the same prescription, by stratifying them into subgroups, you can actually make it so that you only give the drug to the green group who would benefit from the drug without side effects. So if we're going to look at what we do in personalised medicine, I think firstly we should start by thinking about some of the terms we use. So I think most people know genomics and most people know genetics. And these are kind of two of the biggest terms in personalised medicine, where you look at somebody's genome and their genetics to determine their health history. But actually, there's a lot more to it than that. So, for example, epigenomics is, you might have heard of, it's quite a big term. It's basically the protein modifications to the genome. So this can affect how tightly DNA coils together, and that affects access of transcription machinery, which then results in transcription of the transcriptomics. So there's a downward flow of information. This then goes to proteomics. So when we're doing personalised medicine, a lot of the time we will look at the genetic and genomic modifications and then see how that is reflected in protein production. And that's when we get this clinical translation. Another element, which I won't talk about today, but it's really exciting and very relevant, is microbiomics. And that's actually looking at the genetics of all of the bacteria in your body. And that's an entire field all by itself, but we're realising what a massive influence our microbiome on our skin, in our gut, actually has on our health. And there's a lot of evidence that suggests actually the impact is huge. And actually, if we miss that in our genetic screening, that can account for a lot of variability between individuals. So it's also important to understand what genomics actually is and how it differs from genetics. So genetics and genomics are actually two separate fields, although they are interrelated. So genetics is kind of the study of heredity. So for example, if you've looked at these trees in your A-levels or GCSEs, you might recognise them as a family pedigree. And this is when we look at how genes are inherited along families. So genetics is looking at the function and composition of single genes, whereas genomics does look at all the genes, but we also look at the non-coding regions. So these are the regions that affect genetic structure, which again, as previously mentioned, might affect access to DNA of transcription machinery. We might look at, for example, these regulatory regions. We might look at things like the telomeres, which are at the end of the genome, and see how those affect function. So not all of the variation between individuals can be explained by genes alone. We have to look at these non-coding regions as well. And then omics generally refers to looking at the whole of something. So this is where we get into how in personalised medicine, actually, there's a big crossover between medicine and computer science, where because you're looking at so much information, you're looking at the whole genome, for example, or all of the protein modifications to the genome. And if you're looking at the whole of something, you've got a lot of data to work with. So if something ends in omic, it basically just means you're looking at everything in that field. So for genomics, for example, you're looking at the whole genome, the nuclear genome, and the mitochondrial genome. The transcriptomics means you're looking at all of the RNA that is in a cell. So this is what I meant about the downward flow of information. And by actually looking at multiple levels at once, we can see how a change here in the genomics region actually might affect proteomics, or how alterations in microbiomics then result in epigenomic changes. So it's very complicated, but it's basically just very important to know that everything interplays. So a lot of the time, people working in these fields will like to look at multiple levels at once to see how change is carried down. So for example, in my research, I tend to look at genomics and transcriptomics and see how those affect each other. So you've probably heard of the Human Genome Project. It was completed a while ago now, it was in the 90s, and it was a project to try and sequence the whole human genome. And this was a big undertaking because if you were to stretch out all the DNA in one cell, it would actually be nearly 2 meters long. And there's only 4 bases in DNA. So you're effectively trying to piece together a sequence of 4 bases that is billions of bases long. But this has actually massively provided a foundation for all of the techniques we use nowadays, because you hear a lot about next generation sequencing and everyone goes, oh yeah, we can sequence a genome in a few hours now and it only costs a few £1,000s. And that is true. But the reason we can do that is because we have this basis by the Human Genome Project. So if you want to think of it as an equivalent, it's the equivalent of when they completed it at the time. It was like doing a jigsaw without being actually able to see any of the picture. They were piecing it together based on the fact that they knew they'd localized it to a very small section of a chromosome, and then they could try and piece together the overlap. But the reason it took years and years to complete was because they were working completely in the dark. Whereas now, when we use sequencing techniques, we don't need to know where that fragment comes from because we have a reference genome to align it back to. So now we've moved on to wanting to do 999,999 more genomes. And the reason we want to do that is so we can actually start looking at the differences between individuals. So you can see a quote from the premise of the project here, that the basic idea was they wanted to look at variations between cancer patients. So they would take a genome from the cancer and a genome from a healthy cell and compare those and look at how the cancer differed. And then also look at rare diseases. So diseases that have very low incidence in the population, and they would look at the affected patient and their blood relatives and try and see if they could work out the differences between the genome of the patient and their relatives and work out why the patient has the disease when their relatives don't. So as I said, this is a lot of data. It's overwhelming to work with, and the data is usually just stored in what is the equivalent of an Excel document. But again, imagine scrolling through an Excel document that had 3 billion lines. It would be an absolute nightmare to work with. So actually, this is where the computer scientists come into personalised medicine. So computer science is obviously involved a lot of coding, a lot of statistical analysis. And when this intersects with biology, we have a field called bioinformatics. And this is what is used for the bulk of genetic analysis. It's what allows us to, again, align stuff back to a reference genome. So to properly study this information, we use bioinformatics, and we refer to this quite aptly as big data because there's so much of it. You couldn't store it at one person's laptop. It would completely overwhelm the hard drive, and it'd be very unreasonable to ask one person to store that amount of data. So we actually have special facilities where this data is stored on very fast computers. And by actually allowing people to access these computers remotely, this analysis can be done. So let's think about the kind of things we want to do in big data. So one thing you can do is called a genome-wide association study, which basically means we look at the genome and we look at every gene, and then we say, okay, what effect does this gene have? So again, going back to this pedigree, in Mendelian disease, which is a disease where it's being caused by one gene, then basically it's relatively easy to track the disease incidence of that disease. And that's because you can use your pedigree to track affected individuals, track unaffected individuals, and go, okay, this is carried on the X chromosome. But then you might have heard of polygenic diseases before. And these are very common diseases often. So for example, cardiovascular disease is a polygenic disease. And when you look at these diseases, they have a genetic component to them, often quite a significant one. And this is why we care so much about family history when you're a GP. You want to know, does this person have a genetic predisposition to this disease? But we don't often know what the genes exerting that effect are. So how do we find out? If it only increases the risk of disease by 1%, how do we know which genes it is? So that's where the genome-wide association study comes in. And this is basically fancy statistics. So you basically look for these things called single nucleotide polymorphisms, which are just, if you look at this figure here, spots on the genome where one base is different and the bases either side are the same. And these tend to be co-inherited with other SNPs, which is basically, I'm not going to go into it too much because you don't really need to understand it, and it mostly just explains why the statistics work. But if you look at crossing over, it basically means that regions are close together are more likely to be crossed over together. And over time, those regions tend to stay together. So we call these regions loci, and we say, okay, people of these individuals, so people of these loci seem to get this disease slightly more. And when you have a population of hundreds of thousands of people, you can actually start saying, oh, we've noticed that in people who have cardiovascular disease, we see that they have a 20% higher incidence of cardiovascular disease if they have these loci. So that's basically the basis of it. But also you need to take into account lifestyle factors. So looking at cardiovascular disease is a genetic disease, yes, but it's also affected by things like diet. So we know that people who have a diet that's very high in red meat, for example, have a higher incidence of cardiovascular disease. If you have very high saturated fats in your diet, you also have a higher risk of cardiovascular disease. If you have lower rates of exercise, again, high risk of disease. So it's really important to note that in personalised medicine, we aren't just taking into account genetic factors, we also do need to take into account lifestyle factors and actually integrate those in our analysis. And that's where the medics are really important and biologists are really important, because a lot of the time this can be lost if you just have the statisticians and the bioinformatics people working on it. So we also need to make sure we normalise for these factors. So we actually make sure that we do account for them in our analysis, because some factors, for example, might actually be related to genetics. Diet tends to differ globally, and a lot of the information we do in these studies actually differs we have different databases between countries. For example, a real weakness of a lot of data we use is that actually we tend to have collected a lot of it in England, for example, and therefore it might not necessarily apply globally. So this is kind of the process where you basically say, you look at the dots, you say, okay, these dots have a higher incidence of disease. And then if you look at the thing on the bottom, each of those lines where the dots go above that line is a gene that's associated with disease. And then all this information is stored in these databases. So if you look at these databases, there's hundreds of them. There's a database to everything you can think of. Some of them store biological samples where you can actually go and find the original tissue that was taken. And those are often used for things like cancers. So you can actually go and find cancer tissues and analyze them. Q Research is a database that was set up recently and records a lot of information that's been completely anonymized from GPs. And actually, you could do a lot of these databases are publicly accessible. And if you wanted to, you could look at them yourself. I don't know if I'd recommend it. Mostly it's just raw data. But if you did want to have a look at the kind of data that's actually just publicly available, you absolutely can. And this is just great because it just means that we have more and more information available on human health, and it's a real asset of the internet age that actually you can just access this at any time as a researcher. So looking at another case study of how we might use personalised medicine is in diabetes. So diabetes is a disease where your body's ability to produce or respond to insulin is impaired, basically meaning that your blood sugars get high because you can't respond to insulin for some reason. Now, if you look at types of diabetes, the most common types people know are type 1 diabetes and type 2 diabetes. Type 1 diabetes caused by a lack of insulin secretion, and type 2 diabetes caused by a lack of insulin response. But actually, we're going to look at a case study here of neonatal diabetes. Now, this is a rare form of diabetes which has onset when you're a child and usually when you're a baby, hence the name neonatal. And this is a form of diabetes where it would previously have been grouped with type 1 diabetes because these patients don't produce their own insulin. However, it has much earlier onset than regular diabetes, suggesting it was a different disease. And actually, if you look at the genetics of the disease, they identified there was a mutation in a protein channel. And this mutation meant the channel was constantly being held open. And actually, insulin signaling has to be actually stimulated by something. And the scientists realized that, oh, if we can close this channel, the cells actually are otherwise completely healthy. Because in type 1 diabetes, these cells have been completely destroyed. So actually, that's why insulin isn't produced, because the insulin secreting cells are no longer present. But in these patients, they were completely fine, except for the fact that they weren't being stimulated. And they found that when they treated these patients with a drug called a sulfonylurea, the channel actually closed and insulin secretion returned to pretty much normal. And when they looked at the measure of diabetic control in these patients, they actually found out that it was actually better than being treated with insulin injections. They were more responsive to their blood sugar levels and they were effectively back to a healthy level. So this is an example of how actually, if you look at the etiology of disease, you look at what's causing it, you can actually personalise treatment. And this is now the standard treatment for neonatal diabetes. These patients don't need to take insulin anymore. And sulfonylureas can be taken as an oral pill, so it's massively preferable to actually having to inject insulin, which is very uncomfortable. Okay, so I'm going to move on to talking about cancer now, which is my area of research, but I just will pause and see if there's any questions at all.
Hi Holly, it's Lizzie. We have had a few questions in the chat, if you don't mind answering a few of those.
Of course.
Okay, so the first few questions were asking about why you decided to take the time out to do the PhD and what attracted you particularly to personalised medicine.
Sure. So I think I mentioned that I'm part of the Personalised Medicine Society, and we're actually an institution that's based at St Anne's College, along with the Centre for Personalised Medicine, which is the academic side. So that's where a lot of the academics congregate, so a lot of the researchers. And I started at St Anne's in my first year, so that's been my college my whole time at Oxford. Would recommend as a college. But because of the Centre for Personalised Medicine being based at St Anne's, I actually attended a lot of their events, and it's incredibly exciting. I really do think it is going to be pretty much the future of all medicine once the technology catches up to it in many respects. It's also a really fun field, honestly, like you get a chance to try a lot of different skills. So for example, I've learned to code and do a lot of statistics, which isn't something I necessarily would have learned to do in other fields where you don't need that kind of data analysis. So that's kind of what got me involved in personalised medicine. I enjoy the field generally. I was exposed to it a lot. And yeah, I think it's great. In terms of why I decided to take time out to do the PhD, it was a little bit of an impulse decision, I guess. I've always wanted to do cancer research. So I kind of, since first year really, I've been really interested in oncology, even before I was interested in personalised medicine specifically. Although, as we'll see, the two are very interlinked. But yeah, I always wanted to do cancer research, and I'm on a very specific, so I'm on a PhD programme that is specifically focused to cancer research. So as well as doing my own cancer research, I also actually get to attend a lot of seminars with other labs on their research. So the programme I'm on is a new programme. It was actually set on the first group of people doing it. But when the opportunity came up, I was really excited to actually get the chance to work at one of the labs in Oxford and basically just follow a subject I'm incredibly passionate about. And Oxford's really good in terms of they're very flexible on letting you pursue your own interests while you're doing the course. So yeah, it was just good timing, and yeah, they encouraged me to do it.
Great. Oh, thank you. And another question we've got is just about actually a very interesting topic about animal testing. And someone was wondering whether, you know, animal testing was a requirement of research-based courses, and whether that's used across the board or whether it's necessary.
Yeah. So absolutely not, I will say. So the research I am planning to do, I have not started this component yet. I've been doing mostly statistics so far, but it's very likely to involve some degree of animal work. But that is definitely optional, I would say, because there's so many routes you can take your research, and especially on a PhD, it's incredibly self-directed, where you can push the direction of your research in the route you'd like to take it. So, for example, I think it would be nearly unavoidable unless you effectively trained in bioinformatics to avoid being associated with a group that does a degree of animal testing, at least in fields like cancer, because unfortunately, to allow clinical translation, you do currently have to have that precedent of it being effective in an animal model to allow you to implement it in humans. But I will say that if you personally didn't want to do it, you absolutely would be able to find a way not to do it in your research. My supervisor asked me if I was comfortable doing it. And I said I was, as long as I agreed that I thought it was a worthwhile experiment that had genuine clinical potential. But yeah, I definitely don't think it's the only way to go forward in research. And especially as we develop more kind of ex vivo tissue systems. So for example, I'm doing some work on organoids right now, which are an alternative to animal models. And those are great because actually we'll be doing a lot of experiments that previously would have had to be done in mice. And we're able to do them in a way that doesn't actually harm any animals, which is fantastic. The university is really good on the 3 R's, which are basically the principles of reducing involvement of animals and reducing the harm to those animals in research. The goal is to use as few animals as possible at any time.
Great, thank you.
I might take over some of the questions if that's all right. So there's a really good question here actually, which is something I'm quite interested in, is the effects that lack of research into certain demographics. So people of colour we know are underrepresented in clinical research, and sometimes maybe not so much anymore, but women, especially young women, are hugely underrepresented. How does this affect the way we treat medicine? And I guess, is it the same in personalised medicine?
Oh, absolutely. I mean, I think I mentioned before about GWAS databases, and less so with the young women aspect, but definitely if you look at different ethnic groups, it's hugely Eurocentric in terms of the populations. And that's actually a massive issue when you're actually kind of doing a lot of research. There's a real push right now to basically expand the databases to actually include different populations. And again, in terms of what you were saying about young women, a lot of that comes into the risk of them being a pregnant childbearing age. So they're much less involved in studies because a lot of the time they recommend that if you want to become pregnant at any time soon, you don't be in a study. But yeah, I think it's a huge issue. I think it's something that a lot of scientists are actively working to rectify, but I definitely don't think we're there yet. And I think it's really important to take into account if you're doing like a statistical analysis on a population, where you look at the limitations of your population. So yeah, that's a really good point. And I think it's acknowledged, but definitely not at the point it needs to be yet.
Fantastic. And then one last question in this section, if that's all right. So for how often do you need to be screened for personalized medicine? Because wouldn't the body change as you get older?
Yeah, so if you're talking about the genome, assuming you're healthy, remains pretty consistent. You do accumulate mutations over time, but your body will not be overwhelmingly all accumulating the same mutation. So assuming you're doing bulk DNA sequencing, when you're looking at lots of cells at once, your genome will remain pretty consistent over time. Additionally, looking at screening in childhood is predictive of risk of lots of diseases in the future. So again, for that, screening in childhood would be sufficient. Now, if you're looking at things like lifestyle factors, those would need to be taken into account as and when they change. In terms of screening as a whole, I would say that that's not massively something that's currently done without being related to a specific disease. So a lot of screening would be done, for example, if you had a higher risk of a cancer, you would be screened for that cancer. But in terms of screening for your general kind of health, that wouldn't need to massively be done unless there's a significant change, I guess.
Yeah, but I guess what's interesting is that I guess that could be something that would change in the future as maybe.
Yeah, I think definitely we'll hit a point where people will probably just have online health records that they just update constantly, and that will basically be the equivalent of that. But yeah, I think the main place that would come into play is in things like cancers, where you would actually be regularly tracking a cancer. For example, if it wasn't treatable, you would definitely need to regularly screen that because cancers evolve so quickly. But in terms of your genome, that's pretty stable over time.
And speaking of cancer, we'll come back to some of the questions that have come through since then. Thank you, Holly.
Okay. So yeah, personalised medicine is very much already here for cancer, pretty much as much as it can be currently. We already look at cancers to decide therapies. We don't just say, oh, it's a cancer, we'll just give them a generic therapy. Usually there are some exceptions. So for example, testicular cancer will pretty much exclusively be treated with cisplatin or a platinum-based therapy because it's so effective. But generally, the principles of personalised medicine of looking at a patient, sequencing their particular disease, and then changing your therapy based on that is pretty much always already the standard in cancer treatment. So if we look at some kind of how this relates to the traditional model, it's, if you look, pretty similar in terms of we do still give patients chemotherapy, radiation, and surgery. However, now we have all these new therapies coming up where we can actually give much more effective treatments. So rather than giving a general chemotherapy, we can give, for example, a chemotherapy or a hormone therapy which targets that particular cancer. So if we look at a couple of examples, if we look at, let's look at tamoxifen for human estrogen receptor positive cancers and immunotherapy, because those are two examples that are very common. So tamoxifen is an estrogen receptor inhibiting drug. It binds to the estrogen receptor and it stops estrogen being able to access the receptor. Now, in a breast cancer, which is a tissue that can quite commonly express the estrogen receptor, binding of estrogen actually makes the breast cancer cell divide and it stimulates its growth. By having the receptor blocked by tamoxifen, estrogen can no longer access the receptor and consequently the cancer can't divide. Now, there's a couple of ways you can do this. In a non-aggressive cancer, tamoxifen might be the only therapy given and the cancer will die off over time. In a more aggressive cancer, when the rate of division is higher, you might give it in conjunction with other therapies. However, you can give a lower dose of the therapy, which results in fewer side effects, because if you look at actually how chemotherapies work, a lot of them inhibit cell division by, for example, destroying DNA during replication or preventing some metabolites from being produced. But then other healthy tissues also rely on these things, and that's where you get side effects with hair loss and gut damage when you have chemotherapy. So we always want to try and use the lowest possible dose of chemotherapeutics where we can. So by actually testing a breast cancer to see if it expresses the estrogen receptor, we can use personalised medicine to give these patients who are estrogen receptor positive a specific drug that will be effective for them where it would not be effective for an estrogen receptor negative breast cancer. So another example which you might have heard of is actually a chimeric antigen receptor T-cell therapy. And this is a form of immunotherapy, which actually uses a patient's own cells. And the advantage of this is that we don't tend to respond well to cells which aren't our own. We will have an immune response to them. But by using a patient's own cells, you can actually edit them using genetic techniques. So I think if there was a talk on CRISPR-Cas9, that's a technique that can be used to edit these cells. And you make it express an antigen receptor that is specific for the tumour. Now, you would have to test the tumour first to find the antigens that actually can be attacked. But by making it express this, you can actually make the immune system attack the tumour and kill the cancer cells. Our bodies do naturally want to kill cancer. One of the first things a tumour actually has to do is try and escape the immune system. And once it does that, it often does that by hiding all of these antigens. So by actually reprogramming these cells to start them attacking the tumour again, we can generally kill the cancer very effectively. Another important thing to consider that's, I think, the future of our understanding of cancer in personalised medicine is cancer heterogeneity. So currently when we look at a cancer, we tend to just take a biopsy of a cancer in one section and we bulk sequence it. We just lyse all the cells, we break them down, we grab all the DNA, we sequence it and we say, okay, that's what this cancer is. But actually, cancers are evolving things. They are very rapidly dividing and they have mutations in them that mean that they want to have mutations. So our body normally doesn't like mutations. We have a lot of mechanisms in place to repair mutations, to stop them happening. And if they can't be repaired, the cells will apoptose, they will destroy themselves. And if for whatever reason they escape that, the immune system will come along and kill the cells. So our bodies do not like having mutations. However, cancers do. They tend to be pretty genetically unstable. They've generally already lost a lot of their repair machinery due to previous mutations. They're generally escaping the immune system and they're generally supporting themselves. So cancers are evolving pretty quickly. There's a lot of selective pressure on a cancer to try and survive and to try and become more aggressive. And if you look at the structure of a cancer, there's all kinds of cell types in it. So if you look at this diagram here, you can see that actually you've got multiple colours of cancer cells representing different populations of cancer cells. You have fibroblasts, and these are structural cells that are really helping the cancer to actually have a structure. You've got immune cells, and while many immune cells can be bad for a cancer, they kill them, some of them can actually start to become beneficial because the cancer actually convinces them that it's good. You've got blood vessels. Cancers need a good blood supply so they can have enough oxygen to divide. Of course, then some cancers start surviving without oxygen and those become particularly aggressive because actually we rely on cancers needing oxygen for radiotherapy to work. But we've got all these different cell types in the cancer, and this is what I'm looking at as part of my PhD. So I'm looking at these different cell compartments, the immune cells, the fibroblasts and the cancer cells, and I'm seeing how they interact and how they change over time, especially between the precancerous lesions, so the polyps or adenomas, and then the cancerous lesions, which are the carcinomas. So cancers have particular traits that they need to survive. We've already touched on a few of them. We've already said they need to resist apoptosis. We've already said that they need to be able to trigger blood vessels to grow towards them, to give them oxygen. They're tricking the body into thinking they're a useful organ. And they also need to be able to have replicative immortality, which effectively means that cells have an inbuilt trigger, which means that if they've divided too many times, the telomeres, the DNA sequences on the end of a chromosome, they get too short. Those get shorter every time a cell divides. And when they get too short, cells will go, okay, we've divided enough, we're going to stop now. So actually, they also need to be able to overcome that so they can keep dividing forever. And they need to be able to actually evade growth suppressors from other cells. And then they also need to be able to actually, as I said, have more mutations. Having a high rate of mutation is great because it means they're going to start accumulating more and more advantageous mutations. So this is a paper called The Hallmarks of Cancer, and while I would generally not recommend reading a paper before you have to, if you are interested in cancer, I would say this one's definitely worth a look because it has a really nice summary of what cancers are actually like and just like what generally makes cells a cancer versus just a growth. And these are the traits that make a cell a cancer. They mean it's uncontrollably growing. They mean it's not going to be able to be stopped by the body. And actually, we can start looking at these and start actually thinking, how can we target these based on what we know? So we've already talked a bit about the estrogen receptor inhibitors, and this can actually stop cells dividing. We've talked a bit about angiogenesis, and by inhibiting that, you can stop the cells being able to get a blood supply. You can talk about tumor promoting inflammation, so you can use anti-inflammatory drugs. And these are all things that we can use, and we can look at cancer, and by inhibiting it, we can actually make that cancer less aggressive and potentially even kill it. So actually, if we look at these traits, we can look back a bit about what we know about how things evolve. And if we look at evolution generally, advantageous traits, or traits that are useful, are going to evolve multiple times independently. If we look at how animals have evolved, you can see that actually animals that live in the ocean will all develop a reasonably streamlined shape, often with some kind of fin, because this is the most advantageous shape to be in the ocean. And this teaches us that being streamlined in the ocean is good. And cancers do a similar thing. So cancers have lots of different populations of cells in them. And these populations of cells will actually all evolve independently to accumulate advantageous mutations. So we can use a technique called single cell sequencing, where we look at the DNA in just one cell. And if you talk to any researcher, they'll get ridiculously excited about this, because it used to be very hard to do. But you get the DNA from one cell, and you can actually look at how the DNA differs between cells that are next to each other. So the person who asked about screening before, this kind of links back to that. It's really useful to actually screen the cancer over time and see how it's evolving. And then we can look at the DNA, we can look at all these things I talked about before, the genomics, the transcriptomics, epigenomics, and we can actually create 3D maps of cancers. And this technology is still very much being developed. It's very much present in research. We don't commonly do it in actual clinical practice yet, unless it's part of a research study. But by looking at this, we can make these evolution trees of cancers. And what the genes are here is not important. This is just an example of what a tree would look like. But you can see that there's two genes here, in purple and in orange, that have actually appeared multiple times as mutations as this cancer has developed over time. So this is a cancer over time. It started off as this VHL tumour. Again, not important what that means. It's a type of renal cancer. And you can see that these mutations have appeared multiple times. And if we actually look at what these mutations do, this is working backwards. So we found the mutations and now we're seeing what they do. You can see that actually PBRM1, one of the genes, is a tumour suppressor, and ARID1A actually leads to therapy resistance. So we can see that these two genes, when they're mutated, give these cells a selective advantage. These cells are more likely to survive as a cancer. They can resist treatment. They actually mean the cells divide more. So these are great. It's the equivalent of evolution. It's the equivalent of evolving fins, of becoming more streamlined. You also see this if you actually look at germline cancers. So if you know Knudson's two-hit hypothesis, it's basically saying that you need to have 2 hits to a tumour suppressor to actually have a cancer, because if you only lose one copy of a tumour suppressor, the other copy will still be effective. And people who have germline mutations, when it's in the germ cells, so they have every cell in their body is missing one copy of a tumour suppressor, they get these cancer syndromes. And for example is Li-Fraumeni syndrome, which is caused by a mutation in a tumour suppressor called P53. And these people have super high incidence of cancer. They will have multiple cancers normally before the age of 30. Human mixed polyposis syndrome, caused by a mutation in the gene APC. These people will have, again, a very high rate of polyps in their gut. And then BRCA1 and 2, which is a very high, it's a big name, it increases your breast cancer risk. So this is all examples of how mutations in these genes actually massively increase cancer risk. You only need to lose one copy to start cancer developing. So we've talked about this, we've said, okay, there's a selection pressure for certain genes which are advantageous to cancers. So the kind of direction that we're going to take it is we basically want to try and force the tumour to go a certain way. So again, this is what my lab are trying to do. We want to basically apply a pressure to a cancer to make it actually go to a population of cells that we want. So we're going to make that population of cells advantageous to the tumour. We're going to say, okay, we want all of these cells to be green. So we're going to apply a pressure that kills yellow and red cells. And then we've got surviving green cells. Now, normally when this happens, we're killing the yellow and red cells because that's their chemotherapy. And then the green cells become remission, and they will start to evolve more mutations over time. But if we know that the green cells are going to survive, we can actually apply the red-yellow therapy first, and then apply a second therapy that kills the green cells. So this is what the route we want to take cancer treatment. We actually want to start using priming therapies to kill the first population of cells, and then we can attack the second population of cells. And this is, again, how the computer science becomes really useful, because if we know which cells are going to survive, we can do this. Whereas currently we don't understand how cancers are evolving, but we do know the kind of mutations cancers like to have. So by understanding the mutations that cancers like to have, and then saying how a cancer is probably going to evolve, we can actually start seeing how cancer is going to evolve and actually giving them these priming therapies to increase cancer treatment effectiveness. So if you look at it, really, personalised medicine is already here in terms of how we do it. We already screen cancers for certain genes, to see if people have family syndromes and to see if they express certain receptors. In immunotherapy, we already use chimeric antigen receptor therapy. And even in other diseases, we're starting to implement it more and more. People will often be given polygenic risk scores when they have their whole genome looked at and they're told their increased risk or decreased risk of getting a certain disease. However, as we start having these better techniques, as we start being able to look at single cells and DNA, we actually will be able to implement personalised medicine more and more. And yeah, it's going in a really exciting direction, and I think it's a field that's really going to take off in the future. So if you do want to do any more reading about this topic at all, these are some books that I would really recommend if you're interested in cancer or genetics. And if you wanted to go to a talk, see any of the talks by the Centre for Personalised Medicine, they're on the YouTube channel. And there was a really interesting one recently by Professor Jennifer Doudna, who won the Nobel Prize for her work on CRISPR-Cas9. And it's a very good introduction to that. So yeah.
Fantastic. Thank you so much, Holly. That was wonderful. And as you can imagine, many questions are coming through. So I'll just pick out a few and try to, as usual, there's so many, I'm trying to put them together. So I guess it's going back to some of the things that you said earlier on, starting at the beginning. So this might have seemed like a while ago. Immunotherapy that you mentioned can be ineffective against certain cancers, including glioblastomas. And one of our participants wants to know, why is that?
So there's a few components. So firstly, you have to think about actual access of the immunotherapy to the compartment. So some regions in the body have immune privilege, and actually they have very poor access by immune cells. So if you look at the eye, for example, the eye actually, if it didn't have various layers of protection, would be attacked by your own immune cells. Another really good example is the central nervous system, where there's poor access by immune cells a lot of time. So an immunotherapy is going to be less effective on a cancer in a region that has this immune privilege. There's also the fact that cancers often will, as I said, kind of predispose towards immune evasion. And this might mean, for example, that they actually withdraw their antigens. So you might develop a great therapy to attack a cancer because it expresses an antigen, and the cancer, being annoying and also very clever, will go, okay, I'm not going to express that antigen anymore. There was actually a really interesting study on it where they treated a cancer with an immunotherapy with some antibodies, and the cancer stopped expressing the antigen. And then they tested the cancer again, I think like 8 weeks later, and it started expressing it again. So it literally withdrew it while there was a pressure, and then once the therapy had been removed, started expressing it again. So there's definitely tissue considerations and where the cancer is. And you've also just got to bear in mind the fact that cancers are very adaptive, and sometimes just immunotherapy won't work because the cancer outsmarts it.
I guess that's what makes cancer so aggressive. They keep getting away from all of our
They're dreadful.
Oh my goodness. Okay. And then, so are there drugs? So you talked about the microenvironment. Are there drugs that directly target the microenvironment? And I guess we've talked a little bit about the immune stuff, but is there anything else?
In terms of in cancers, so to a degree, so I talked about anti-inflammatory drugs. An anti-inflammatory drug for a cancer would not be targeting the cancer, you would be targeting the microenvironment. Again, angiogenesis, so VEGF inhibitors, which inhibit angiogenesis, those would also be classified as targeting the cancer microenvironment. So those would definitely both be classified as microenvironmental therapies. I think there's definitely potential for more of it. So there's a real actual growing interest in cancer-associated fibroblasts, which is a specific type of fibroblast which seems to kind of develop in a cancer. And I think there's going to be potential for targeting those in the future as well. But a lot of it just relies on actually just targeting the things that help the cancer survive, sort of passively targeting the microenvironment.
Fantastic. I think Lizzie has a question for you.
Yeah, sure. So we got a question earlier on just asking what your kind of day-to-day life is like as a PhD student.
Yeah, it's not necessarily entirely representative currently because there is a pandemic, but I tend to kind of try and keep my schedule pretty 9 to 5. Especially after the medical school, where I think it is very easy to get caught up in a lot of the work, and I think you definitely don't need to. It's very possible, and I think extremely healthy to have a good work-life balance. So I mean, I tend to have a pretty normal morning, get up, breakfast, work 9 till 5. I tend to have quite a few seminars and meetings because part of being a PhD student is you're doing your own research, but you're also trying to learn, and you're trying to, go to things and learn new things and think how they can apply to your research, which a lot of the postdocs and a lot of the people who run the labs don't really have time for. So PhD students are often the ones who go to teaching at the university and say, oh, I went to this cool seminar and I think we could use this technique in our lab. So yeah, and then I do a lot of work for the Personalised Medicine Society, as I have mentioned multiple times, would recommend the society. And we run like coding workshops and try and really increase access of the skills you need to kind of do this analysis in personalised medicine. Yeah, and I also do a class at the language school. So I do a Japanese course every week, which is great. And it's a really good way to kind of like unwind while still working on something fun. So yeah, I'd also very much recommend the language school if you have any interest in languages. The classes have been online during COVID, but they're still really good.
Oh, that's nice to hear that you've got a nice, well-rounded work-life
balance. I feel like
balance.
it's really important to have a good work-life balance. I really value it.
And then just one last question, asking, do you think that AI technology could help predict cancer evolution?
Oh, absolutely, it is. That's a big part of what we use in it. So if you look at kind of how you look at cancer, how cancers evolve, it's neural networks and it's people trying to develop better algorithms to actually study this. So yeah, I mean, I think if you look at the people actually who are doing a lot of the bioinformatic work, a lot of them either come from an AI background or are developing AI software, or so yeah, I think AI is a huge part of personalised medicine and it's only going to become bigger. And if you have an interest in AI, I would absolutely recommend you look into personalised medicine, because I think it's a really nice way to kind of integrate a lot of this with real kind of medicine and human aspects of the science. Yeah, a lot of people working in AI are going into tech. But I think if you don't like tech, personalised medicine is a hugely underrated field in AI.
Okay, fantastic. Okay, so I think in the interests of time, we might call it there.
Yeah.
If that's all right with you, Holly. Thank you so much for your time and for your wonderful talk. We will be posting this on YouTube, on our YouTube channel, and also on our Instagram page. We'll post about the Hannah Weinberg paper, because that was actually one of the first research papers I ever read. And it's a review. It's a review, so it's not as heavy as an actual paper. And there's a nice summary.
The introduction. It's really good if you just want to look at like what cancer is and how scientists like to talk about it.
Okay, fantastic. Thank you all, and hopefully we'll see you all again next week. Thank you again, Holly.
No problem at all.